Okay, so today we're going to talk about protein synthesis. So what I want to do is show you this video here, and it's going to kind of go through it, and I'll stop it. So what we're looking at right now is
the basic unit of all living tissue.
The cell, obviously.
There is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes.
We talk about the genome, we're talking about all the genetic material, right? We've said that every organism has a complete set of instructions on how to make a whole U, and these sets of instructions, although usually loosely defined throughout most of the cell cycle, at some point become wound up tightly and structured together into these packages called chromosomes.
Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes.
Okay, so you're not going to have to know all of this, like what a histone is and things like that, but it's good to hear the terminology. It's good to know some of the things that are going on. The important thing is you have this section of a chromosome, and look at the way they had the picture here. They kind of start to unwind this chromosome and show you this long strand, and a chromosome is nothing more than a tightly wound strand of DNA. But different sections of it are the genes. And all that means is that there's a specific section of a chromosome that is the set of instructions for a very particular thing. Could be your height, could be your weight, could be how tall you are, could be your skin color, could be whether you have an attached earlobe or not, could be whether you have hair on your fingers. Very specific traits. Okay, so when we talk about how we inherit certain traits and things from our parents, the set of instructions that code for those proteins is the gene, right? The reflection of who we are protein, collections of proteins. And so the instructions for making these proteins are the genes.
These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus.
So you don't really have to know RNA polymerase and this and that, but do know that when a cell wants to make a protein, this is what happens. We've talked about the fact that even though every cell in your body has a complete set of DNA to make a whole new you, different cells specialize. They use only the instructions they need for the purpose they serve. For instance, an eye cell is not going to unravel the DNA for heart muscle tissue. Your toes are not unraveling the DNA for making a finger. Okay, so every cell specializes in what it does, depending on the type of tissue it is and the organ it is within. So when it happens, though, a copy gets made. It's like if you went to the library and the library had a very rare book. The library may not want to lend out that book and let it out of its house, out of the library to give to you. But the library may be very comfortable with putting the pages you want to read on a photocopy machine and making those copies and then sending that home with you because it knows it still has its original. This is kind of what happens with DNA. So what you should have learned right now with DNA is that the base pairs A, T, G, and C, adenine, thymine, guanine, cytosine, are the nucleic acid base codes for DNA. And as you can see on this side of the strand, that's all you see. These free nucleic acid bases, they come from the food you eat, right? They're one of the 4 major organic compounds that we've learned about: nucleic acids, lipids, carbohydrates, and proteins. And so these nucleic acid bases come from the other foods you eat and get broken down, delivered to your cells through your circulatory system, and then, as needed, get reassembled to the things you need. So the only difference between a strand of RNA and DNA, one of the major differences, not the only, but the major differences, are the code differences. If you notice, there's an A, there's a C, there's G, but there's no T. In place of T, there's a U, standing for uracil. There is no thymine in RNA, only uracil. So whenever there's an A on DNA, it will attach to a U, right? The rules for DNA are A binds with T, G binds with C, and that would be the case for our double-stranded DNA. So here's the unzipped DNA. This over here would be the T to go with that A down there. But when we're making a copy of RNA, there's only a single strand, and there is no thymine. So whenever there's an A U will be put in its place. By the way, you're going to hear in a minute, but this process is called transcription. We're transcribing. If I put a set of notes up on the board, you would make your own copy of those notes. You would be transcribing, copying down what I'm telling you. And maybe every other word, wherever I put the word the, you might put the word and, right? Because, you know, you're using you instead of my code, right?
The DNA code determines the order in which the free bases are added.
I'm just stopping for a minute so you can see the different codes. Again, no thymine in RNA, so there's a U. But when there's a C, there's a G. A, U, G, C. There is A, there is adenine. So when I have a thymine on my DNA, it becomes adenine in the RNA molecule. Okay?
The messenger RNA. This process is called transcription.
So this copy we've made is called messenger RNA because it's going to send a message to the rest of the cell about what particular protein to make. This is all happening within the nucleus of the cell right now. But in order for a protein to mate, if you remember, we've got to involve the ribosomes because ribosomes make proteins. So watch.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
It's going out of the nuclear pores, the small holes in the nuclear membrane, and coming through those pores out into the cytoplasm.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acids. Transfer RNA molecules carry the amino acids to the ribosome.
Okay, so that was a lot really quick. So there's another type of RNA called transfer RNA. And transfer RNA's job is to pick up loose amino acids. Again, where do these amino acids come from? They come from the proteins that you eat, right? Amino acids are the building blocks of proteins. So when you eat beans, or when you eat a hamburger, or you eat some kind of food that has proteins in, you digest it, you break it down to the small amino acid molecules. There are 20 of them that we know of in life. And your body will then take those amino acids and synthesize them together, which means to make their proteins, to make human body proteins, okay? So each amino acid has, each transfer RNA has a very specific shape to pick up a very specific amino acid, okay? And so what you're going to know is that every transfer RNA has this called a anticodon. Notice it's three nucleic acid base pairs long. This is called an anticodon. And it's going to bind to the code on the messenger RNA.
The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
Okay, I was trying to catch it when it wasn't blurry. So you see what's happening here. The amino acids are just coming in where they belong. If you look at the codes here, here's the messenger RNA, the message, okay? And the message here is GAA. Well, it will only allow an anticodon of these 3 base pairs attached to a transfer RNA that has the opposite that will attach. So in this case, G pairs with C. There is no thymine, no T in RNA. So A binds with U, and A binds with U, and U binds with A. G binds with C, U binds with A. And so only a very specific transfer RNA will come in at any particular 3 base pair codon sequence. And it will only be able to attach because it has the proper anticodon. So they're not really showing this, but the ribosome also has specific shapes to allow, a couple of transfer RNA in at a time. And as soon as the amino acid gets delivered to the new ones coming in, right, these amino acids get linked together. We're building a protein. And these transfer RNAs are then released to go back to the cytoplasm to pick up amino acids again. And this is happening thousands of times every second of every day. You're constantly making proteins that you need to build structures, to replace worn out cell organelles, to send chemical signals through your body, to do all of your metabolism, right? All of these chemical processes are known as your metabolism.
This is added to a growing chain of amino acids.
One more time, as soon as you link 2 amino acids together, you have a protein. But you can also have a protein that is thousands upon thousands of amino acid bases long.
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Proteins are not single lines of amino acids. They are made of these amino acid molecules, and they will interact with each other and form different types of covalent, ionic, and hydrogen bonds that will give them specific shapes. And that's what causes them to function. So let's just talk about a real life example for a minute. When we're talking about, COVID-19, right? COVID-19 is able to attack your cells because it has a specific shape to allow it to attach to certain protein sites on cells, allowing it to enter the cell. So proteins have shapes naturally in your body that do different things. We call it lock and key model. If I've got some kind of substrate, some kind of chemical in the body that looks like this, I might have a protein, a specialized enzyme that looks like this, that has the complementary shape. And so it allows it to act and do things to that substrate, either break it down or bring other substrates in to build more complex molecules. And that's pretty much the end. That's protein synthesis. So just to look at, I've got a little thing here just to show you real quick, little slideshow. And it's going to be very, very short.
Molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is
Okay, so just to look at the DNA code, A binds with T, adenine binds with thymine, guanine binds with cytosine. And that code tells the cell what proteins to make, what type of trait to produce. And a trait is a characteristic. So it could be something as small as a protein within your body, or a physical appearance, right? So this isn't showing the whole thing, but 2 things have to happen. You have transcription, where we take the DNA sequence and make an RNA messenger RNA. Again, the difference is that RNA does not have thymine, so you have uracil in its place. When that message comes out into the cytoplasm, it's picked up by a ribosome, and now we have another molecule coming into play, transfer RNA. The transfer RNA picks up the amino acids that you get from the digested food you eat that are delivered to your cells by the circulatory system. And now these transfer RNAs with the anti-code, the opposite code, will attach in the appropriate place on the messenger RNA, the 3 base pairs, that is the code or codon. And notice they're opposites. If I have UUC on my message, U binds with A, U binds with A, C binds with G. I've got AAG for the anticodon message. The amino acids keep getting delivered to the one that the new transfer RNA coming in, and the ones before it get released back into the cytoplasm to pick up amino acids again. So just a quick recap. Here's a little graphic organizer showing you a flow chart. DNA provides the code to form messenger RNA. Messenger RNA attaches to the ribosomes. Transfer RNA reads the messenger RNA. Amino acids are added to the growing protein chain. So if you were looking for a very simplified idea, that's it. Okay, just to show you again, RNA includes things like messenger RNA and transfer RNA. And what are their functions? Well, I know one of the functions is to carry amino acids. And one of the functions of messenger RNA is to copy the coded message from the DNA. Messenger RNA also carries the message out of the nucleus to the cytoplasm, to a ribosome. The transfer RNA is carrying amino acids and then adding amino acids to the growing protein chain, okay, based on the proper sequences. All right, I hope that was helpful. I hope you have a good understanding of RNA protein